Can you see now? It's better? Okay. I don't know what happened. I did get a little symbol that said my quality was bad. I wonder if like my people logged into their classes in the rest of my house. Okay. So it's not a circle. So what is the shape of the DNA that you're going to draw on the nucleus? Okay, it's linear. And what is it, a straight linear line or is it squiggly? Okay, what kind of squiggly? We actually gave it a name. What's the name of the squiggly that makes the DNA look like? A double helix. That's exactly right. A double helix. So it has that very consistent squiggle shape. Excellent. And you're done. You did it, right? So now you should be able to answer this question. What four structures can be found in all cells? You already did it, right? DNA, cytoplasm, ribosomes, and cell membrane. We're going to talk about those all again in just a second. Now let's think about this. What are some things that all cells, we know they all do? Every cell has to do it to survive. Okay, they have to grow and use energy. So that means they have to make organic material into ATP so that they can reproduce. Okay, Rithik, good. They have to make proteins. What's that process that we talked about called that ends up making proteins? The central dogma. Every cell does the central dogma. It doesn't matter whether you're prokaryote or eukaryote, animal or plant. You all do central dogma. You all make ATP through cellular respiration. All cells have to reproduce. Fabulous. Can anyone think of any other processes that have to happen? So all cells have to interact with their environment. So they have to exchange things across their membrane, oxygen and carbon dioxide and sugars and salt, all those kinds of things, right? So we'll talk about this semester too. All right, real quick, let's review. The plasma membrane is the outer boundary of the cell, right? So the function of the plasma membrane is to be selectively permeable. What that means is it allows water, oxygen, carbon dioxide to go in and out of the cells as needed, all right? It is selective as to what can go in and out. So we'll talk next chapter about what can go in and what has to have special help to go in and what will never go inside a cell. All right, we talked about the components of the plasma membrane. What are they made up of? It was from the last chapter of the last section. What are membranes made up of? Phospholipids, right? So now you should probably picture what that looks like, and you can see it in this picture, right? But it's got the polar head, right? The glycerol that's connected to the phosphate that's connected to this choline, and then two tails. And that it always situates itself into a bilayer. So that's what makes this so unique, that it situates itself into this bilayer where the tails face each other and the heads face outward towards water. So there's water on the outside of the cell, there's water on the inside of the cell. So it makes this sphere where all the heads are facing out or all the heads are facing in, okay? Now, in addition to phospholipids, membranes also have other structures. So you'll find proteins embedded in the membrane that serve many different purposes. So you have proteins that maybe are channel proteins, other proteins that are enzymes, other proteins that are receptors or signaling proteins. And you can even find some carbohydrates hanging out at the cell membrane. So a lot of those molecules we already talked about are very important. What's unique is that every cell has a membrane made of phospholipids, but the combination of proteins and carbohydrates that are found are what make each cell's membrane unique, which is pretty cool. All right, so those purple structures you see in this have to span, for example, the hydrophobic area of the tails and the hydrophilic area of the heads and the water. So let's think about this. Let's apply what we've already learned to the fact that these proteins have to be embedded in the membrane. So this red ribbony thing is our protein. You'll see the alpha helix structure here, right? It spans the entire membrane. It has parts that sticks out, other parts that stick out down here, parts that are in the middle, okay? So this is a protein. So what are the building blocks of proteins called? Amino acids. And tell me again, what are the two functional groups that are on every amino acid? Amino and carboxyl. So in this picture, you should probably recognize the amino and the carboxyl. Here's that central carbon. Here's the amino. Here's the carboxyl. Hopefully you recognize that. This is the R group. All right, so maybe think in your head a little bit what kind of R group this must be. So is this a polar R group, a non-polar R group? Is it possibly acidic or basic? Okay, I'm getting some different answers here. Gracie, why did you say polar? What made you think polar? Okay, remember this nitrogen and these oxygens kind of cancel each other out. So when we categorize them, we only categorize them based on their R group. So we're not going to look at that nitrogen and oxygen. Okay, so the answer is this is a non-polar R group. In the R group, from this point down, there are no oxygens or nitrogens. So given knowing that, here's my protein. Where would we find that amino acid? Would we find it in the A region of the protein, in the B region of the protein, or in the C region of the protein? And you're right, Noah, it would have basically like a neutral charge, right? It wouldn't be acidic or basic. It would behave in a neutral way. So where do you think you would find it? A, B, or C? Okay, we got an A. Anyone agree with Miles? So Miles, what made you say A? Yes, C, I think C is probably in the cytoplasm, and A is on the outside of the membrane. So where would we find the amino acid at the top of the page, that phenylalanine? Would we find it in region A, B, or C in this protein? And Miles says A. Miles, why did you say A? Can you give us your reasoning? You think it may be, you wrote GOB. So Anna had a good suggestion. Think about phospholipids, think about how proteins fold, and then think about that R group. Okay, so here's what I'm going to tell you. For sure, you're going to see this in B, right? You're going to see hydrophobic nonpolar R groups where you have those nonpolar tails of the phospholipids. If you're facing out to the watery solution of the cytoplasm or the outside of the cell, you're more likely going to find one that's polar, right? So what about this amino acid? So again, here's our amino group. Here's our carboxyl group. This is my R group. So is this amino acid polar or nonpolar? Okay, for sure this one's polar because we've got the oxygen and the nitrogen. So where would we find it? Do you think we'd find it more along this part of the protein, or the middle? C, good. So facing the cytoplasm. A, good. So either of these places you would see it sticking out because it would be able to interact with the water here. Excellent. Does anyone want to just give a guess? What do we call this structure here in B, that helical structure? Okay, it's secondary structure. That's right. It's a kind of secondary structure. Does anyone remember the name of it? Alpha helix. Good job, Gracie. It's an alpha helix. And the reason I'm pointing this out was I just asked you last time what the shape of DNA was, and that was a double helix. I want you to make sure you keep those straight from each other. And I'm going to show you a double helix again today, so you'll see it. But remember that they're both helical in that they twist around like a spring. But one is, the word double helix is associated with DNA, and the word alpha helix is associated with the secondary structure of a protein. All right, so those are words that sometimes, like on a test, they can be very easily confused with each other. So I want you to make sure you keep them straight. Okay, we have cytoplasm. What kinds of things do we find in the cytoplasm of a cell? Any cell. I'm full of questions today because I feel like you already know what the answers are. Okay, but good answers, but not any cell, right, Stephen and Gracie? What kinds of cells would have organelles floating in the cytoplasm? Eukaryotic, right? So the first question there says, what do we find in the cytoplasm of any cell? So you're answering the third question, right? Okay, ribosomes, good. And that's really all we've learned so far, not DNA. Because in a eukaryotic cell, the DNA is specifically inside the nucleus, right? So not DNA. In a prokaryotic cell, yeah, you would find it. So in any cell, the only thing we really learned about that's in the cytoplasm so far are ribosomes. But you would also find sugars and proteins and things like that dissolved in that cytoplasm as well. Now, there are some things that are uniquely found in the cytoplasm of a bacteria that's different than what might be uniquely found in the cytoplasm of a eukaryote. And you guys already mentioned it. Uniquely, you would find DNA in the cytoplasm of a bacteria cell, which you would not find in a eukaryote. However, in the cytoplasm of a eukaryotic cell, you would find organelles like the nucleus and the mitochondria and the ER that you wouldn't find in a bacteria cell. So make sure you can distinguish one from the other. Okay, so inside every cell, we also have DNA, right? So we now know the structure of the DNA is the double helix. Looks like this. Remember, it's two strands that twist around each other. And they form a chromosome. So does anyone know what the word chromosome really means? Or what it's made up of? You can kind of see in that picture, it's labeled. Okay, it's made of genes, right? So what happens is, if we were to take the DNA out of just one cell of a human, and we were to take it and line it up, it's really skinny, but if we were to line all the DNA up end to end, it would be really long, a couple feet, okay? So we have to fit a couple feet of DNA into a really small, not just cell, into a really small nucleus, right? Really, really small space. And keep it organized, because if we have it really crazily unorganized, we can't access it to do the central dogma, which is so critical, right, for everything. So the DNA is organized in a very specific way. And it's kind of like, I'm going to pull, let me see if I have one here without unplugging, without using my my cord here for my speakers. So it's kind of like if you have a big long cord, if you wrap the cord around like this, you keep the cord organized, right? So that it's not going to get tangled or anything like that. Same thing like with a vacuum cleaner. You know, the back of the vacuum cleaner, you can loop the cord around so it's not dragging on the floor. It organizes it. It makes it take up less space by looping it around. All right, so DNA does the same thing. It's super long. So what we do is we wrap it around proteins to kind of condense it to a smaller space, just like you would wrap your cord around the plug part. And the most condensed version that it becomes is what we call a chromosome. Okay, a chromosome is very, very condensed DNA that's wrapped around proteins. So in a way, a chromosome and DNA are analogous in the sense that the same DNA that's in your cell is found in the form of a chromosome where it's really condensed. All right. So DNA and chromosomes contain genes. So does anyone want to define what a gene is? Anyone know what a gene is? I know you know. Okay, perfect answer. A functional segment of DNA, a sequence of nucleotides, two very good answers. And what does the gene code for? What are the instructions? Okay, for a trait, and traits are then dictated by protein. So you guys, that was fabulous sequence of answers, right? It's a functional sequence of nucleotides in the DNA that code for a protein that gives a particular trait. Great answer, right? And that is the whole idea of the central dogma, right? That we have regions of DNA that we copy into this middleman mRNA that we use then to make into the protein that then functions in the cell as a trait or some sort of enzyme or something like that. So let's answer this last question. So then what part of the DNA do we actually copy into an RNA? Do we copy the whole chromosome? What part do we copy? Okay, half of it. You might be thinking of like half of the double helix, like one side of the double helix. Is that what you mean, Stephen? Okay, you're right. We're only going to copy one strand of the double helix, but how long is it? How do we know when to start and stop? It's the length of a gene. So if we go back to this picture here, we see that this is my big long double helix. We have from here, these nucleotides, remember these are nucleotides, ACGT, ACGT. From here to here are those instructions to make one protein. This is what we're going to make into RNA, just the length of a gene. All right, so genes are what code for proteins. And genes are just sequences of DNA. And DNA is compacted into chromosomes. So in a way, these are very much related. I mean, in a way, these words are sometimes used interchangeably, right? Ashley, did you have a question? Yes, no, maybe so. So when DNA is copied, it's not just one long strand of DNA, it's segments of a gene. Okay, so that's a really good question. So when it's copied, we copy the entire length of that linear piece of DNA. So in humans, we have 46 linear pieces. Each linear piece in a human has about 1,000 genes on it. Okay, so when we copy it, we just copy a little teeny portion of it. It's kind of like if you were going to go to a cookbook, you wouldn't aim to make, scalloped potatoes. You wouldn't copy the whole cookbook to make scalloped potatoes. You would just go in and copy that recipe that you needed, right? So think of it like that it's the chromosome itself has all of the instructions that the cells need, all of the chromosomes together, right? In terms of any protein it might have to make. But when we just need one protein, we're only going to copy one gene at a time. Okay, great. All right, so remember, we're going to really talk about the central dogma over and over. There is an overarching emphasis on the central dogma when it comes to cell structure. So I really want you to pay attention. And in particular, we're going to pay attention to what kind of protein we're making, because the different kinds of proteins take different pathways through the cell to get to their final destination. So we're going to talk about that. They know which side to know. There's kind of like an on-ramp to the gene, and so the proteins that are doing this know how to get on the on-ramp and then move forward with it. So they're going to know which gene on-ramp to take. And when we get to transcription and translation, we'll talk a little bit more about that. Okay, all cells also have ribosomes, which remember are protein factories. This is where we make the protein. Now, ribosomes are a compilation of proteins, so they're called ribosomal proteins, and RNAs, they're called ribosomal RNAs. So we mentioned that there are different kinds of RNAs in the last chapter. In particular, there is this RNA that we just saw with this dogma that's called the messenger RNA that carries the message from the gene to the ribosome. The RNAs that function in the ribosome are called ribosomal RNAs or rRNAs, okay? And they never get made into proteins. They function as an RNA. So we'll talk more about functional RNAs a little bit later, but it's important that you know. There are other functional RNAs that help with the whole protein making process called transfer RNAs or tRNAs, okay? So at the ribosome, you have this crazy compilation where the mRNA is bringing in the code from the gene, the rRNA is helping it line, and then the tRNA is helping convert it into amino acids. And then the proteins of the ribosome are holding it all together, okay? So we'll talk more about ribosomes in this chapter, and we'll actually talk way more about rRNAs and tRNAs when we get to later in the semester when we get to transcription and translation. Okay, so let's answer some questions. What are ribosomes made of? Go ahead and put it in the chat, because we don't have colored cards. And I didn't make polls. Is it RNA only, RNA and proteins, proteins only, DNA and proteins, or is it DNA, RNA, and proteins? Okay, got some B's. Good. Ribosomes are made of RNAs that are called rRNAs and proteins, that's right, which are called ribosomal proteins. Okay, let's answer this question. Where are functional ribosomes found in cell? This is a little bit of a tricky question. You might know something about this. We haven't really talked about it yet, but this is just kind of a little foreshadowing. Does anyone know where functional ribosomes are? So that means not while we're building them, but when they're functioning, where do we find them? Okay, cytoplasm and ER, great. That's right, we have them floating cytoplasm functioning. Some of them are bound to the ER functioning. Now, we're gonna find out that ribosomes are somewhat assembled inside the nucleus, but they don't function inside the nucleus. So that's why the question asked about functional ribosomes. So make sure you know that even though they get assembled in the nucleus, they don't function until they get out to the cytoplasm. Excellent, good job, guys. All right, so we've already talked about this. If we were to take all the cells in the world, we would know they had those things we just talked about, and then we could categorize them based on how they're organized. What are the two different types of cells? The two different organizational plans. You can cheat and look at the picture to the right. It gives you the answer. Yeah, prokaryotes and eukaryotes, right? And we know that they are the same in that they have plasma membrane, genetic information encoded by DNA, cytoplasm, ribosomes. They all do the central dogma. They all do cellular respiration. Every cell does those 6 things, right? And then we have these differences, right? The differences between prokaryotes and eukaryotes, such as in a prokaryote, the DNA is a circle. In a eukaryote, the DNA are these linear pieces. In a prokaryote, there's no internal compartments. In a eukaryote, we have all of these amazing internal compartments called organelles. A prokaryote is very small because it doesn't have any internal compartments. The internal compartments of a eukaryote make it so it can be bigger. Okay, so make sure you can compare and contrast what's similar and what's different between these two types of cells. All right, so let's talk for a minute about prokaryote cells. We've talked about them already in lab, so this shouldn't be anything really new to you. We know that they don't have organelles. So in particular, they're often referred to as not having a nucleus, but they don't have any other organelles either. They don't have an ER or Golgi or mitochondria, right? They just don't have any internal compartments. They got their name because they didn't have a nucleus, because that was so obvious under the microscope. They have DNA that's circular, right? And then you may hear, and I don't really love this word, I wish it kind of went away, but it's just not my call. They wherever the DNA is floating in the cytoplasm is called the nucleoid, right? I just think it's just a terrible word. Why does it have to have a name? But it does. But it's not membrane bound, so it's not an organelle. Okay, we know prokaryotes are small. How small is small? What's the range of size for a prokaryote? Good, 0.2 to 2, not nanometers, Brianna. What's the right unit? Micrometers, that's right, micrometers. Nanometers, remember, are like the DNA and the protein. Okay, great. They're ancient, right? They have been on Earth for about 3.8 billion years, right? They are 1 billion years in age older than us in terms of eukaryotic cells. They are ubiquitous, they're everywhere. There's good bacteria, and then there's bad bacteria. They're the ones that make us sick and the ones that don't. They're single-celled, although they very much like to sometimes hang out in a group and share duties, like we saw with the Anabina, right? So they're pretty unique. They're very simple structurally, right? They don't have a lot of internal structures. All right, so let's look at, I know the other thing I want to say. Bacteria can be heterotrophs, which means they have to go find organic food to eat. They can be autotrophs, which means they can do photosynthesis. They can also be decomposers, which means they can take dead organic matter and do something with it. So they fit into all of the different categories of nutritional food web. All right, so if we were to look at a bacteria, we would find the things that we know all cells have: plasma membrane, cytoplasm, ribosome, DNA. And then we would see sometimes some additional structures. So we're going to talk through some of those: the cell wall, a capsule, flagella, or pili. Okay, so let's start with the bacteria cell wall. All bacteria, well, 99% of them have a cell wall. What is that cell wall made of? So we've learned about a lot of these different structures. D, good job, peptidoglycan. So that's a word you're going to be able to want to remember, all right? And this peptidoglycan layer can be thick, a real thick mesh-like structure, or it can be thin. And that helps us determine one kind of bacteria from another. The ones that have a really thick one, what do we call that? Anyone remember from lab? Gram-positive, good. And the thinner ones are gram-negative. And we can just do a dye process to tell the difference. All right, so is the cell membrane different than the cell wall? Yes, all right. So let's review what they're different, how they're different. So are they different in structure? Yes. One is made of phospholipids, which you see right here. The other is made of peptidoglycan, which you see up here. Totally different. Phospholipids are a lipid. Peptidoglycan is a sugary protein mix. Totally different. Right now you know the difference between organic molecules. All right, what about in function? First, let's review. What's the purpose of a cell membrane? We went over it like 3 slides ago. A little bit protection. It does provide some protection. That's true. That's not the main purpose. Okay, to hold the stuff in, that's true too. Right, but its real function, and the reason why it's maintained being this structure for 3.8 billion years is because of the fact that the interior is nonpolar, where those tails are, and it is selectively permeable about what it lets in and out. All right. So if it's selectively permeable, that means it is like a gatekeeper that's letting some things in and some things out. So now let's talk about the function of the cell wall. What do you think the cell wall's job is? Hold on, I think I don't know how to see private messages. Someone just sent me one, but I don't know how to see it. So if you were the person that sent it to me, I'm so sorry, but I can't see it. Okay, so you're right. The cell wall helps maintain structure, helps maintain rigidity. It gives us rigidity, right? It's a protection. Let's see. So I have to go where the people are. I don't even know who it was that did it. I just saw it pop up and then it went away. It came up again for like a split second. Tune in chat messages. Click back on everyone. So where everybody's name is.
So at the top, if you're in the chat box, if you click the little arrow next to where it's everyone, it'll take you out of the chat from everyone.
Oh, yes, I see it.
See your private messages.
Okay. Okay, so Alexis, I've seen your message, so make sure you just go back and watch the recording for that part that you missed. Okay. All right, thank you, guys. I didn't know how to see that. I should, and I really should know how, but I didn't know how. Okay, so that's the difference. They're different structurally, they're different functionally, right? So you should be able to, again, compare how are they the same. They're both boundaries for the cell, right? They both are kind of found in the same place, right? They're on the outside of the cell in a sense. However, a cell wall doesn't touch cytoplasm. It's only in the extracellular space, where the cell membrane touches both, and then they perform different functions. Excellent. All right, in addition to a cell wall, bacteria cells can have a capsule. So a capsule is not for every cell, okay? It's just some cells. And the capsule is a thick layer of polysaccharides, all right? So it's kind of a sticky, ooey gooey layer. Not every bacteria has one. In fact, this can be used to determine the likelihood that a bacteria might make us sick or not. So what do you think? Do you think the ones that have capsules or the ones that don't have capsules are the ones that can make us sick? Okay, good. The ones with capsules. So the reason for that is that they get into our system and our immune cells are like, yeah, you're foreign, we're going to get you. And they go to get them and it's like Velcro or like glue and they get stuck to the cell, but they can't do their job in terms of getting rid of them. And so our immune system is a little bit slowed down by this sticky, ooey gooey layer. You know what it makes me think of? You know, in Home Alone, when he puts like the car on the steps and the guy's trying to go up the steps and he loses a shoe and then he loses his other shoe and then he loses a sock and then he loses his other sock and it takes him forever to get up those steps. That's what I think of when I think of a capsule. You're slowing down the immune system so they can't do what they need to do, and it's allowing the bacteria to flourish during that time and make more of itself. All right, so that's why it's considered a virulence factor. The ones that don't have capsules, they get into our system, I'm breathing them in right now, and my immune system's like, oh yeah, we know you're foreign, and it's completely getting rid of it immediately before it ever has a chance to divide and make me sick. Okay, so that's a capsule. The other thing we find in bacteria are flagella. Those are long hair-like structures that you can find. Here's one on, this is, what shape bacteria is this? I'm not really sure that's this kind of bacteria, but if this were a bacteria, what's the shape of it? Because really these are the little bacteria right here. Oh no, not Coccobacillus. The one on the left, I'm looking at the one on the left where my little pointer is. Do I need to make my pointer bigger? Let's see if I can make it bigger. Okay, spirillum. So you can't see my pointer. Let me see if I can make it bigger. I don't know how to make it bigger. Oh, I can highlight though. This one, right? This one's the one I'm looking at. So you can see that this one has a tail here and it has a tail there, right? Those are flagella. Here's one. This one is Coccobacillus. That's a really good description of that one. And you can see it has 3 tails coming off the back of it. So the flagellas are like tails. They really function in movement, right? Now, flagella are not unique to bacteria, right? Can you think of an example of a eukaryotic cell that has a flagella? A sperm, exactly. So functionally, they serve the same purpose. A flagella helps a sperm swim. A flagella helps a bacteria swim. Structurally, they're a little bit different. So in a bacteria, and you do not have to know this picture, but in a bacteria, it's made of a different kind of protein that sticks out of the edge of the cell. It's anchored in a different way, okay? And we'll find out that a eukaryotic one looks very different than that. All right. Also, bacteria cells can have these shorter hair-like structures called pili, or sometimes called fimbriae. Fimbriae is also the word, if you ever took an anatomy class, that is, are the finger-like structures that the fallopian tubes use to hold on to the ovary. So you might have heard that word somewhere else. And their main job is to attach to a surface, attach to another cell, all right? In particular, when they attach to another cell, so you can see this here, this is their attachment right here. This is a long pilus that's connected them. This is called a sex pilus because in biology, the word sex means that you're combining DNA from 2 different sources, right? So for example, when our skin cells divide, that's asexual because there's not a mom and a dad, right? But when we reproduce, it's called sexual because we're combining DNA from mom and combining DNA from dad to make a new organism, right? So because this is one kind of bacteria here, and this is another kind of bacteria here, and this little tunnel forms, and part of their DNA from this cell travels to this cell, now all of a sudden, this is a combination of 2 different kinds of bacteria, right? Which is kind of cool. This is actually how bacteria cells can very quickly spread antibiotic resistance. All right, so let's just say we have a population of bacteria cells like this, and one of them has inherited a mutation that allows it to be resistant to an antibiotic, right? If that one cell shares its little piece of DNA with its neighbor, and then the neighbor cell shares it with its neighbor and has babies, right? The ones that have that little red piece of DNA are going to survive. The ones that don't are going to die off from the antibiotic. But the next population of bacteria that's in us, the ones that survive, all have that resistance, which means now if we take that same antibiotic, it's completely ineffective, right? This is why you don't take antibiotics unless you really need to, one, and unless you know for sure you're sick with a bacterial infection. Okay, so I'm going to get on my soapbox for a second because this is one of those things that I feel very strongly about. First of all, antibiotics completely changed the lifespan and survivability of humans. So when antibiotics were first discovered, people that would die regularly from what seems like common diseases were not dying from them. They were able to live through them. Polio, chickenpox. People used to die from chickenpox and measles and the flu, right? People die. People still die from the flu, right? But the flu is not a bacteria, but anyway, so you would die from all of these things. And so what antibiotics did was antibiotics were able to kill the bacteria that was causing some of these diseases, right? Not a virus-based disease, but a bacterial-caused disease. And so people that would die from them would all of a sudden live, right? So antibiotics were like a complete game changer. okay? Same thing with vaccinations. So antibiotics helped us survive through bacterial infections. Vaccinations help us survive through bacterial infections and viral infections, right? So now, if you go to the doctor and you're like, oh, my throat hurts, I think I have strep throat, right? Strep throat, we learn now streptococcus, right? We know that's a bacteria that's causing strep throat. But if the doctor just says, okay, well, I'll give you some antibiotics and go home and take it, you'll feel better in a couple days. If what you had, if your sore throat was not caused by bacteria, you're taking an antibiotic and you're basically causing evolution and natural selection to happen within your body's bacterial flora, right? So all of the bacteria that are in your body can die from that antibiotic that you take. Well, the ones that won't die are the ones that are going to be antibiotic resistant. So let's just say you had a viral sore throat. You were going to get better in a couple days anyway. Our bodies are really good at getting better from viruses. But you took an antibiotic and now you changed the population of bacteria in your system to have more antibiotic resistance because the ones that survived through the antibiotics that you took have the resistance in them, and they had babies and they passed it on to their neighbors, right? So when you do have strep throat, and you know when you have strep throat, by the way, you wouldn't go to the doctor and say you have strep throat because you would be like passed out on the bed because you couldn't move. Strep throat's like the flu, like you get completely knocked out. The doctor would say, oh, I did a culture. You definitely have strep throat. I saw streptococcus. Maybe he even gram stained it to prove it to himself, right? Now he gives you the same antibiotic and you go home and you're like, I am not feeling better, right? And what happens is that antibiotic that he gave you is not effective because you've changed the bacteria in your system. So if you take antibiotics, or if a doctor says to you, take this antibiotic, say to them, are you sure I have a bacterial infection? Because I absolutely think if you have a bacterial infection, take it. You don't want to die from this, right? But if you don't have a bacterial infection, there's no reason to be taking an antibiotic if you just have a virus. And so you might be thinking, well, why would a doctor do that? Well, unfortunately, we have like some of those Care Nows, and I go to Care Now if I'm sick and it's the middle of the night too, right? But they are for-profit institutions, so their job is to make money. And the way they make money is that you come and you pay a copay when you get there. And so they want you to come back to them. So they want you to think they made you feel better. So if they give you medicine and then you feel better in a couple of days, they're going to think, oh, Care Now took good care of me. I'm going to go back there next time I feel sick, right? However, if you had a virus, you were going to get better in a couple of days, regardless of whether you took an antibiotic or not. So they didn't really make you feel better. Your body made you feel better, but you feel like, well, they wrote me the prescription for the antibiotic and that made me feel better, so I'm going to go back to them. And so there's a little bit of method to that madness, right? They're making money off of it. So you, as informed citizens now, can make that distinction for yourself, and you can say, wait, stop. I want you to do a swab. And sometimes that little swab costs you extra money. Like they actually charge you more money, like $12 more to swab your throat to make sure it's a bacterial infection. But I always make them do it. Like that's what, two meals at McDonald's, I can handle it because I want to know for sure that it's strep throat and not just some viral thing. Okay. So Sim asks, is it possible if children take heavy antibiotics? I don't know that there's any really solid data supporting that. I will say if you take really heavy antibiotics anytime in your life, whether you're an adult or you're a child, you run the risk of, because we have a lot of bacteria in our system that's good for us, like helps us digest foods and all that kind of stuff. So if we take heavy antibiotics and we kill some of those good ones, sometimes that allows some of the bad ones to flourish, and that can be a problem. So unless you really need to, you shouldn't take super high doses of strong antibiotics. So yes. So if the doctor gives you an antibiotic prophylactically because you had surgery, absolutely you should take it, because you definitely don't want to get an infection where you had surgery, because that could lead to like sepsis. So absolutely, there's a whole reason why they give that to you on that reason. I mean, they opened up your body to the air where there are bacteria, and they don't know what landed in there. Sterile as they can be, they can't prevent all the bacteria from falling into your wound, right? So they are prophylactically giving you that. So just to combat, if anything did fall into your wound, you won't get a bacterial infection that would cause possibly sepsis. So can I tell you a story? All right, my niece knows that I tell this story because she's like your age. She's, I guess she's a senior at UNT right now. But when she was like 16, she got her wisdom teeth out, just like Gracie. This is not my niece, Grace, though. It's her sister, Chloe. And they did the same thing, Gracie. They gave her prophylactic antibiotics so that she didn't get a bacterial infection in her wisdom teeth sockets. Well, she was not feeling very good, and she spiked a fever even after they gave her that prophylactic antibiotic. So they went back to the doctor, and the doctor said, you know what, I think maybe it didn't work. Maybe that wasn't the right antibiotic. I'm going to give you another one. And they gave her a really strong antibiotic that kills lots of different kinds of bacteria. So kind of one of those wide-ranging antibiotics. And so she took that for the 10 days that she was supposed to. And she really wasn't starting to feel very much better, but it was more of an intestinal thing. She was starting to have really bad diarrhea and kind of stomach cramping. And then one day, and this is like she's 16. How many 16-year-olds look at their poop in the toilet? I don't know. But she looked and she realized that it was bloody. And so she went to her mom and she said, Mom, I think that there's something wrong. I haven't been feeling good and I have bloody diarrhea. And so they took her back to the doctor, and it turned out this 2 rounds of antibiotics within a month, she did 2 10-day rounds kind of right in a row, killed the good bacteria in her intestines. And one of the things, I mean, the good bacteria help you digest food, but they also help you keep the bad bacteria at bay so that they can't overproduce, okay? So she killed out the good bacteria, and a bad bacteria called C. differens flourished, all right? And she was not feeling good because the bad one was not good for her and causing her to have very bad stomach cramping, bloody diarrhea, bad, right? So they sent her home and they said, take probiotics and eat yogurt, right? You need to re-flourish your good bacteria in your system. She didn't get any better. She was still sick, still pooping blood. And so they went to the doctor a couple days later and said, she's not feeling any better. Her fever is just not going away. You know, this isn't working. So they told her that she had to have a poop transplant. It's really called a fecal transplant. You may have heard of this before. Basically, what they needed to do was squirt somebody else's poop up into her intestines, and somebody else's poop had healthy bacteria in it that would possibly reflourish her intestines. So they told her to do this, and so her dad pooped. Her mom took the poop and kind of mixed it up in kind of a milky solution and then enemaed it up into her butt, right? And thinking that this was going to solve it. And at this point, she is like super sick. I mean, it's been 20 days of her being like really sick. And she doesn't get any better. In fact, she gets worse. They have to put her in the ICU. And like my husband and I, who are biology teachers, were like, oh my God, she has a C. diff infection. Like people die from C. diff infections, right? So we're like thinking, oh my gosh, Chloe like might not make it. Like this is our 16-year-old niece and we're worried about it. So they admitted her in the ICU and they said, we're going to give you another fecal transplant. That's really the only thing we have to help you right now. We cannot give you any more antibiotics for sure. And we need to get rid of this C. diff. We need to get it under control. So this time she had a random donor that still gave her poop, and that one worked. It finally started to work. And so she had to have 2 poop transplants. And really it was just kind of about, I mean, she didn't abuse the antibiotics, but the antibiotics were what caused all of that to happen. So I suggest, like, I hardly ever let my kids take antibiotics. Like, they have to be really sick before I give them antibiotics, just because I saw this happen to my niece and it's scary, right? But on the flip side of that, if my kids were really sick, I wouldn't hesitate in an instant to take antibiotic. If they had surgery, I would still give them an antibiotic because I wouldn't want, I mean, Chloe's situation was pretty rare. That doesn't happen. People take antibiotics all the time after they have surgery and they're fine. It just so happened in Chloe's case that it wasn't so fine. So that's why your moms are probably like that. They may have heard these same kinds of things. And your moms and dads, well, they're probably a little bit younger than me, but they know that antibiotics used to be like this major lifesaver, and now all of a sudden they're not as effective. And so they're seeing that transition to abusing it, and really Care Now kind of places, I feel like they're like antibiotic pushers, like drug pushers, because what they do is they provide you with an opportunity to abuse it and not necessarily educate you totally about when and where you should take it. Because, I mean, even I've gone there and they've said, well, if you don't feel good in a couple of days, I'm going to write you this prescription and take it. They shouldn't let that be up to me. If I don't feel better in a couple of days, I should go back to them and they should reassess me and they should make sure that it's bacterial before they just give me an antibiotic, right? I never fill those prescriptions that they hand me. So that's why your parents are like that. Probiotics are interesting to me. If you think about what happens in your digestive system, it's hard for me to imagine that the probiotic bacteria make it the whole way down to your intestines, because they're going through all of these harsh environments and all of this like degradation process. I mean, your whole immune system your digestive system is built to like not let bacteria get the whole way there. Now, obviously, at some point during your life, you had to ingest those bacteria to get there, but I don't know how probiotics make it through. Like, it's an interesting conundrum to me that I've never looked up in research, so that's why I don't know the answer to it. Okay, so I'm really going off topic, but that's okay. It was fun. I got to tell you a poop transplant story. I'll have to call Chloe today and tell her that I told her poop transplant story. She's come to TCC before, and so with me, and so my students have been like, "Oh my God, that's the poop transplant girl," and she's like, "Yeah, that was me. I'm the poop transplant girl." Okay, so every bacteria cell also has DNA. Remember that it's in that place called the nucleoid, but it's not membrane bound, so it's not an organelle, right? Bacterial DNA is a circle, not linear. It is associated with protein, so it does condense and kind of twist up, but it's still, if you were to untwist it, it would be a circle, not a straight line. So here's a good question. Does a bacteria DNA have genes in it? What do you think? What do genes code for again? A trait, and what causes traits? Proteins. So do you think bacteria have traits and proteins? Yes. So would they have genes in their DNA then? Yeah, exactly. They have genes. They just have less genes than we do. They have maybe, 5,000 genes and we have 30,000 genes, right? So we just have a lot more than they do. Excellent. All right. So this is just I stole this. This is actually someone else's PowerPoint slide that I found online, but I liked this picture just to kind of show you how the association with the protein works in the bacterial chromosome. So this would be down here. This is your chromosome. It's a circle, right? And like I said, it kind of twists up. So what happens is there are proteins right here. These are DNA binding proteins. And they bind to the protein and cause the protein to loop out, kind of like a flower. It's a very pretty looking little flower, right? And then those are further twisted into this, what's called supercoiled DNA. And that's how it can be very, very condensed and inside the cell. And so what we're going to find is sometimes DNA needs to be loosey-goosey, and then other times it needs to be really tight. And the cell controls when and where that happens. And so it knows that, if we have to do the central dogma, we need it to be a little bit more loosey-goosey. if we have to divide the cell into two, we want it to be really tight so we don't lose a piece, right? So the cell makes a decision on how much of the coiling that happens. Okay, so that's our bacteria cell. Any questions on bacteria? Okay, so next we're going on to the eukaryotic cells. We know that eukaryotic cells have linear DNA, that DNA is found within a nucleus, that we have a whole bunch of other kinds of organelles we're going to learn about, like the mitochondria and the ER and the Golgi and the lysosome and the peroxisome, right? So we're going to learn about each of those. Let's remind me, what's the size of a eukaryotic cell? Much bigger, right? 10 to 100, what's my unit? Micrometers, excellent. And notice how much more recent they are in terms of the age of the Earth, right? Big Bang was 4.6 billion years ago. We went a billion years without any cells. That took some time to organize. And then we finally got cells, and we went another 1.5 billion years before we got to eukaryotic cells. All right, so that's pretty far along in the process. In terms of their internal structure, they're very complex in comparison to the eukaryotes. So they have these internal compartments that compartmentalize function and make things happen differently. And then remember, we have in the eukaryote domain, we have three kingdoms in a group, right? The animal kingdom, the plant kingdom, the fungus kingdom, and then we have that last arm of the tree that is our protists, which is just very diverse, and there's lots of controversy over calling it a kingdom, okay? So if we were to take a look at the inside of a eukaryotic cell, we can see immediately how much more busy it is. Filled with stuff, hardly any cytoplasm really. It's just filled with all of these organelles that are all doing various different functions. And so our goal in this chapter is to learn what all of these different parts are doing. That last picture was an animal cell. This one's a plant cell. So we're also not only going to be able to differentiate between prokaryotes and eukaryotes, but between animal and plant. All right, because there's some distinctive structures that you find only in one or the other. All right, now we're going to break this down into processes. I'm not really big on just learning the name of something and recognizing the shape if we're not relating it to what the cell's actually doing. So we're gonna focus on the process and then talk about the parts that participate in that process. All right. So I kind of broke it down into 4 processes. So first is manufacturing, right? And that's basically the central dogma. We're going to be manufacturing proteins, and a little bit we're going to talk about manufacturing lipids. If we're manufacturing, that means we're building. So what kind of process are we using if we're building things? What's the process called where we build stuff? From chapter 4, condensation reactions. Good. Dehydration synthesis reactions. Excellent. So manufacturing is going to involve the nucleus, because that's where the genes are in the DNA. So hopefully you already knew that. It's going to involve ribosomes, right? Because that's where the proteins are going to get made. And then we're going to find that some proteins also have to go to the ER and to the Golgi to be finally processed. Now I have the word ribosome in purple and the other words in red. What do you think the difference is? Why do I have ribosome in purple? What do you think? How does a ribosome different from a nucleus or an ER or a Golgi? It's functional, it makes proteins. Anybody want to take a guess? Okay, good. It's in both prokaryotes and eukaryotes, so we can't call it an organelle because we know prokaryotes have it. What makes it not an organelle? What did I say has to be true of something that's an organelle? It has to have a membrane, that's right. And a ribosome does not have a membrane around it. So we don't consider it an organelle, or at least a membrane-bound organelle, right? So that's why it's a different color. that's going to help you later. Okay, the next category of process is breakdown of molecules. So what's the process we use to break down molecules? Hydrolysis, great. And that's done in two different organelles, the lysosomes and then the vills, so we'll talk about those. Then we're going to talk specifically about the process of energy processing. So the mitochondria, does anyone remember what's the process that the mitochondria does? Okay, it makes ATP, it's the powerhouse. Anyone know what the name of that process is called? That's right, cellular respiration. So we're going to learn a little bit about mitochondria and cellular respiration, and then we're going to do a whole chapter on cellular respiration. So we're coming back to that one. And then what do the chloroplasts do? Photosynthesis. That's exactly right. So those are both energy processing parts that a cell has to do. And then the last category is structural support. So giving the cell structure. Movement, and so this is kind of two kinds of movement. So the cell can move, but then also things within the cell can move. All right, so movement of components and movement of the cell itself, and then how the cells communicate with other cells. All right, so those involve things like called the cytoskeleton, which is our basically like a protein scaffolding, the plasma membrane, and the cell wall. All right, so they're purple. So what does that tell you? That they're not considered organelles, right? They're not considered membrane-bound organelles either. So we know the cell wall in a plant is made up of cellulose. That's a polysaccharide. The cytoskeleton is a bunch of protein. Okay, so my time is officially up. So we will stop here. We will start with manufacturing tomorrow night in lecture at 6:30. And I'll go as far as I can, and then whatever I don't do, I'll record and I'll post it after. Okay? So good job today, guys. You guys did a lot of answering questions, and I was proud of that. Any questions for me before we stop? Backer.
This is regarding the discussion questions on
Okay.
So one of them is why is water essential to life, and then it lists like all the terms. Do we have to like say all the terms that are in the question like in our answer?
Well, you have to say the ones that are relevant. So the ones that I've listed there likely are going to be relevant. So probably a good idea to list them.
Like to list all of them, like each one?
Well, there might be some that are not relevant, but if they are relevant, yes, make sure you list them.
Okay, no problem.
Any other questions? Do you feel like the discussion questions helped you with the test material? Yeah, that's the whole point. It's to force you to study, right? To make you answer those questions, which helps you study. So good. And it's fun that you get to choose. My husband gave his test yesterday, and they were given 11 questions ahead of time. They could answer all of them, which they should have. And then they had to show up at 6:30 last night, and he picked 4 of them. They didn't know which 4 he was going to pick, and then they had to write their answers and turn it in right that minute. So if they didn't do it ahead of time, then they had to kind of come up with an answer on the spot. So I kind of like the way we did it better, but it's a neat way to do it. Okay, guys, well, I'm going to be here for office hours, so feel free to stay. Otherwise, email me if you have questions. I'm going to stop this recording.